Okay, so I'll skip slightly back then. Doing the brief version. So I'm Verneri Anttila. I'm a research fellow here at Mass General and the Broad Institute with Ben Neal and Mark Daly. And Dennis asked me to give a slightly cheeky version of the presentation, of basically doing the other side of the argument on the common variance side, now that we've heard various things that we can do on the rare variance side. And so the argument between common variance and rare variance as explainers of common disease has been going on about 100 years. So, and we had Pearson and the biometricians on the common disease, common variance side, who suggested that you should have many different variants of small effects to explain why we have various phenotypes in the population that behave in a normal fashion. And then Bateson and the Mendelians on the rare variant side, who basically had a version of the argument that that instead of having one unified disease or one unified cluster for the disease, each disease is actually a number of various different kind of unique groups, each mimicking the same phenotype in some way. And so basically what follows from this model is that if we look at the common variant side, the sort of logical development of that proposed by Fisher was the so-called infinitesimal model, in which basically everything in the genome could have an effect on each phenotype, and you basically have a normal distribution of effects, and once you get across a particular threshold, then you get that particular disease. That basically there is so much give in the system, and once you go past the amount of give, then bad things start to happen. Whereas the rare variant argument is that basically there's many different ways of breaking the system, or which are unique. So I like to call this the Tolstoy model of disease, in the old quote of how all happy families are the same, but all unhappy families are unique. So recently there was a very nice paper from the Type 2 Diabetes Consortium, published in Nature, where they evaluated in a comprehensive fashion all the variants identified in both common variant and rare variant approaches within the disease, and basically then plotted the mononuclear frequency of each variant against the odds ratio to basically show what the distribution of the variants looks like, as a sort of measure of basically how have evolutionary forces affected this particular phenotype. And so we can see here that the TCF7L2 variant, which has been known for a very long time, is the sort of lone outlier. But basically everything else, all the way from the 50% mononuclear frequency to 0.1%, follows that sort of distribution there, which you can see of basically small effects across the board. And as we know, there are a number of examples which we all know by name of such alleles as the TCF7. For example, ApoE in Alzheimer's disease, that sort of have these outsized effects. But those, on the other hand, are the alleles that have been recognized early on in research, because the statistical power needed to identify a variant that explains here roughly 1% of the liability scale variance is very low, so these are very easy to detect. But basically what their paper made the case for is that as we move down the frequency spectrum, there doesn't seem to be as rapid a rise as the rare variant side of the argument would perhaps be hoping. That basically you seem to be having a very modest increase in the observed odds ratios at lower frequencies. To sort of flesh out the story further, so Robinson et al. In a couple of years ago published a paper which looked at the distribution of a similar distributional picture of the common disease variants reported in the GWAS catalog up to that point. And you can see that if you squint your eyes pretty hard, you can sort of guess at a similar distributional behavior as in the previous picture. Of course, the GWAS results typically end at roughly 1% of the allele frequency, but you can sort of squint your eyes hard enough to see this kind of same kind of flute shape in both quantitative and complex traits. Perhaps more in the quantitative end, but there's still a case to be made for similar behavior across the phenotype spectrum. In the same diabetes paper, they had also some nice simulation work to basically see what the data that they have amassed would look like under different models of sort of relative importance of rare variants versus common variants. And as you can see here, so there's three different models looking at rare variants are mostly driving diabetes, then a 50-50 model between common and rare variants, and then a common variants are primarily driving the disease. And you can see that the empirical distribution in type 2 diabetes is looking much more supportive of the common polygenic side of the simulation, suggesting that the existing variants that they have captured in their data set is indeed behaving in the sort of expected manner under the common variant hypothesis. So then talking a little bit about the common variants and why this is the case. So in recent years, we have seen a rapid increase in the number of genes identified for various phenotypes. I'm showing here two of the success stories in the neuro and neuropsychiatric space, namely schizophrenia and migraine, where we have a fairly steady and also predictable increase as sample sizes go up. The number of loci discovered also going up. So the schizophrenia one has a bit of a kink in the graph, given that I'm only listing cases and not controls. So there's a considerable difference in the number of controls between the 21,000 points there. But you can see that as you pile a certain number of additional samples, you will see certain number of additional loci in a very nice fashion. And so the reason this has happened in the last couple of years is that first of all, the methodology used to do quality control and to sort of evaluate the data has been standardized to a very large degree. So we have standardized solutions for how to deal with the data to a much larger degrry than sequencing still has. This might be changing in the next probably year or so with software like Hail coming out, but still that's currently the case or a reason why the GWAS variants are easier to come by than the rare variants. Further, the limiting step for the frequency that the GWAS data can interrogate has been due to how large is the reference size for the imputation activity. So for the GWAS data, we use a method or an approach called imputation to infer what the rare variants near the directly genotype variants are based on the haplotype patterns from reference sets compared to the disease set of interest that you're working on. And we have seen in the last few years a rapid increase in the amount of reference data available, namely with the Haplotype Resource Consortium that published the paper this summer, showing that that with the 32,000 samples in the reference panel, we can get the reliable imputation estimates to a much lower frequency. So you can see here on a solid red line, the sort of high confidence calls at 0.8 of average aggregate R square, and then what the current standard is, for example, within the Psychiatric Genetics Consortium and the UK Biobank is a average R square of 0.6. And when we compare this to the diabetes paper recently, we can see that with the more, first of all, here at 1% is the sort of typical traditional lower end for GWAS. At the solid red line, we can see the where we can interrogate on aggregate at 0.8, which means that we have a very good chance of capturing the vast majority of the variation at this threshold. And at the currently used lower bound, we can go slightly lower than what the diabetes paper could evaluate variance to. So basically what we have here is that the space between where their sequencing experiment is and the GWAS experiment is narrowing. So basically together we're starting to be able to capture a much larger chunk of the variance space, because of course the sequencing experiments are also growing in size. So basically they will be approaching us from in this region quite well. A few words on the technology and the benefits that the chip technology has is basically what the sort of decreasing prices and the availability of combining the data with existing data sets, or the new genotype data with existing data sets, gives us a possibility to drive down the prices for these chips. So basically at the sort of prices that we can do these days for a genotyping chip, we're able to, the field is rapidly moving towards large-scale biobanks, which we're able to genotype at much more advantageous prices as before. So for example, here is the current few top contenders in the biobank space. So UK Biobank, which many of you are probably familiar with, who have about 500,000 samples in their database, with the final batch of genotypes slated to be released this fall. There's a Chinese biobank of similar size, and of course the commercial one by 23andMe, as well as several other research databases in Helsinki, California, and Vanderbilt. So basically, that on the sort of common variant side, we can expect that the sort of massive number of samples, together with unified medical record usage in the next years, will yield a lot of very interesting findings. As I mentioned before, so one of the other advantage is that you have large disease consortia which are rapidly able to genotype a lot of samples, which then consequently allows you to go further down the frequency spectrum. So for example, here I'm showing the PsychChip consortium, which we're currently working on here, led by Pamela Slar and Ben Neal, where we have about 200,000 samples done within the last roughly last year and a half, which is sort of the kind of sample approaches that you can rapidly get when everybody's using a standardized platform and done in a centralized manner. Of course, many of these approaches give you then the sort of downstream analysis that you can do are things which we can do with multiple modalities. So here on the left side, I'm showing the Psychiatric Genetics Consortium's common variant pathway analysis results from a paper from last year. And on the right side, Menak and Frommer's paper on similar psychiatric networks from sequencing side of things, which was already mentioned by a previous speaker here. So that basically there are many of the things that you would like to do then post analysis, like Dennis's paralog score, can be done with both common variant and the rare variant side. However, one thing I would like to bring up as a difference between these, which is something that I feel is key for understanding, is the total variance captured by things of different frequencies. So here's the same graph that, or same figure that Andrea showed earlier, where we see the sort of a relative contribution of, or relative amount of variance captured by the different methods, where we can see that the polygenic risk score here is capturing a lot more of the total variance in the in educational attainment than the rare variants are, which makes sense because while we know that there are more rare variants than common variants, most of the variation in the genome does live in the common variants. And so here's a way to illustrate that. So this is from a paper by Alkas Price's group from last year, where they looked at contributions to heritability by different minor allele frequencies. And as you can see, both in the simulated data here, where alpha shows different behaviors for simulated effect sizes, and then in the real data from the schizophrenia consortium, that you can see that the higher minor allele frequency bins capture a lot more of the variation than the lower frequency ones, and this line will keep on going down the lower you go in the allele frequency space. And as we know here, that of course the amount of variance captured by each bin is directly related to our power to detect changes within the variance of that bin, and also further that the heritability that we measure is also a direct arbiter of whether variants are important. So basically, the more a single variant can explain of the difference between cases and controls, the more important that variant is likely to be in the etiology of the disease. So here I'm showing as an example, so the schizophrenia and migraine data that showed before, that how are we doing in terms of capturing that heritability then. So here in schizophrenia, we can see that we're roughly capturing 26% of the total heritability, and the most reliable twin estimate we have places the total heritability at 61%. So the middle bar here shows what based on the current data, when we project the sample to, or rather, Paul Rouleau from Alkas Price's group has projected what the GWAS heritability based on our current understanding of schizophrenia be at an infinite sample size. We're roughly capturing a little bit over 50% of the heritability here. In migraine, we are roughly about a third of the way in there, where we haven't done the projection yet, but it still gives you a measure of roughly how much of the variance we can currently explain with the data. And as was mentioned by the previous speakers, so on top of this, CNVs explain roughly a little under 1% of the data, and the ultra-rare variants also explain another roughly 1% of the data. So basically, that gives you an idea of how we're doing in the chase for the missing heritability. One of the other cool things that we can do with heritability, given all the power that we can do with the heritability that we can capture from the common variants, is that we can do heritability analysis directly, which has recently been boosted by the introduction of LD score by Brendan Bullock-Sullivan and Hilary Finucane. And one of the cool things that they can do, which I wanted to bring to your attention, is that the amount of, once you have enough of the heritability captured by the GWAS data, you can start partitioning that heritability into different subgroups. So here, for example, Hillary's paper from last year, where we showed that we can start asking questions like what kind of genomic marks, and also what kind of tissue distributions we can divide the heritability in. So basically asking the question of all the variants that we are capturing for, let's say, schizophrenia, in which tissue and which genomic marks does that live. And this gives us a very nice approach to refine our understanding of the phenotypes, which we couldn't do with if we didn't have this much of the variance captured. Another example of what we can do is here from Elise Robinson's paper from this year, where we looked at the correlations between the score for autism and various other measures within the neuropsychiatric spectrum. So, for example, we could easily, with the heritability approaches, we can compare how much of the correlation to the variance that explains autism in our data can be correlated with various other things, such as social and communication difficulties in children, and then also other types of psychiatric susceptibility. Further in work that we're that's currently in progress to be submitted, what we can look at is also the how does the comorbidity and correlations between the different brain disorders behave. So here we can see an analysis of how much the heritability for one particular brain disorder is correlated with the heritability for another disorder, where we can see that in psychiatric phenotypes we largely see very highly degrees of correlation, whereas in neurological disorders, including epilepsy, we see a more specific type of heritability. So that basically, on average, a variant that increases your risk for a particular psychiatric disease has a higher chance of being also risk causing for another psychiatric disease, whereas in the neurological space, we can see that the risk profiles seem to be reasonably private to each of the diseases, and with very little significant sharing between the phenotypes. Further, we can then break this down into components of other measures. So basically, we can ask what the college attainment and educational scores that Andrea was showing earlier, that how well do these correlate with various brain disorders. And here you can see, for example, that the psychiatric diseases, or several psychiatric diseases, tend to be strongly correlated with increased college attainment and increased years of education, whereas the several neurological disorders are associated with lowered college attainment and education. And we can expand this this kind of analysis to as many modalities as basically we have phenotypes for. So we can do metabolomic analysis, we can do imaging analysis, and many other types. So basically, that was my sort of my case to be made for why common variants should not be forgotten. So why it's important to do essentially both. So there's many things, of course, where variants are also very important, because, for example, the two models proposed originally propose a different kind of way of breaking the system in the etiology of the disease. So basically, we do need to evaluate both methods. But for example, currently our laboratory here at the Broad is investing now heavily into generating a lot more common variants than rare variants in the for the new Common Disease Consortium, which hopefully will get around 1 million samples going by within the next couple of years. So that basically the case is made here for the importance of both ways of looking at this data. Thank you. That's what I had. And are there any questions?